Hi, my name is Connor Ruen. I'm a first-year master's student here at SDSU, and I'm majoring in plant science. My project is working with living mulch, of which we are working with 3 different clover varieties: red clover, cured clover, and white clover, along with a bare ground treatment. And within those treatments, we have 4 management treatments, which are no-till, tilled, tilled fabric, and no-till fabric. The goal of the project is to prevent nitrogen leaching through the soil profile, prevent soil erosion, along with increasing organic matter and fertility of the soil. A cover crop is a crop that is grown when there is no cash crop in the field and it is in fallow. This is usually planted in late fall and then grown throughout the winter into the early spring before being tilled under, crimped, or terminated with a herbicide treatment. A living mulch is a cover crop that is grown concurrently with the cash crop and covers the ground to prevent the soil from being exposed to the environment. The cover crops that we are growing are 3 different varieties of clover that are all perennial. So this is the second year of the study that I am working on, and it was established the previous year by Alexis Barnes. Since I am working on the second year of this treatment, I am looking more into the soil health aspects and the benefits that these living mulches can have on the soil. One of the soil health aspects that I am looking at is infiltration rates within the no-till and tilled treatments. Within those treatments, we notice there is a very large increase in the no-till water infiltration rates compared to the tilled. In one of the plots, the no-till treatment had nearly 2,500 milliliters of water infiltrate the soil in the first minute, in comparison to most tilled treatments having 500 to 700 milliliters of water infiltration in the first minute. So there is anywhere from 3 to 5 times as much water infiltration in the first minute compared to no-till to tilled. To collect the infiltration data, we take a 10-inch PVC pipe that is about 6 inches tall and pound it 2 inches into the ground using a 2x4 and a rubber mallet. Once it is sunk firmly into the ground and we are confident that it will not move or leak any water out the sides, we will begin to pour water into the ring to a designated line, and then every minute we will refill that water to that line and then take the amount of water added to calculate how much water has infiltrated the soil profile per minute. Another soil health aspect that we are looking at is soil compaction. The way we take the readings for soil compaction is by using a penetrometer, which is a metal rod that you press into the ground, and it will take a reading of how much pressure it takes to penetrate the soil profile. Once the penetrometer reaches a certain threshold, you can stop and measure how far into the ground it is to find a limiting layer or compaction layer, also called a hard pan, or you can sink it to a designated depth to see how much pressure it would take for a plant root to penetrate through that soil to that depth. In my half of the study, I'm looking at how the cover crops and living mulches affect the yield of broccolini, which is a crossbreed of European broccoli, which is the broccoli most people think of, and Chinese broccoli, which is another variety of broccoli commonly grown in Asia. The crossbreed of those 2 species is what we call broccolini and has a long, slender stem that is sweeter and more tender in flavor. I'm growing 2 purple varieties of broccolini, which are the Bernarda and Burgundy varieties, and then 2 green, which is Melody and BC 1611, all of which I have sourced the seed from johnnyseeds.com. We planted the broccolini on June 30th into the greenhouse and then transplanted all those seedlings into the field on August 9th and expect to have a harvest by the end of September. Some of the data that we will collect from the broccolini once we are harvesting is the shoot length as well as the biomass of the mature plants once we are done harvesting. This will be taken to see if there is a difference in the stem length before the flowering. This will be done to compare how the different treatments affected the size of the plants as well as how large the marketable product will be once it is matured. I'm currently using row covers to prevent any cabbage looper butterflies from laying any eggs on my broccolini, as well as preventing any animals from getting to my broccolini and eating them before I am able to take harvest data in the future. Some of the cool things I've noticed in my fields is that there's an overabundance of pollinator species that are often visiting the nectar-rich clover flowers as they are popping up throughout my treatments. It is really cool to see the different pollinators that are coming and visiting my plots, and I hope to potentially do a little study in the future on the different species that are coming to my plots. I'm continuing research that was originally funded by USDA Specialty Crop Block grant and is administered by the U.S. Department of Agriculture and Natural Resources. To learn more about this research and other projects in our lab, please visit the SDSU Extension website.